in a groundbreaking development that was previously deemed impossible. Scientists have used a speck of mouse brain matter the size of a grain of sand to create the first ever precise three-dimensional map of a mammal's brain. This map, which reveals the intricate details of neuronal connections, provides unprecedented insights into the brain's structure and function. This remarkable achievement was accomplished by an international team of 150 scientists from 22 institutions led by the Allen Institute for Brain Science, the Baylor College of Medicine, and Princeton University. The project, known as the Machine Intelligence from Cortical Networks Microns Program. Their work was recently published in the journal Nature. Using advanced imaging techniques, the researchers meticulously recorded the brain activity within a 1 cubic millimeter portion of tissue in a lab mouse's visual cortex. The area responsible for processing visual input. They kept the mouse awake and visually stimulated during imaging by having it run on a treadmill while watching 10-second movie clips, including scenes from The Matrix and Mad Max, Fury Road. Clips of extreme sports like motocross, luge, and base jumping were also included to keep the mouse engaged. Once the imaging was complete, the mouse was euthanized, and the same cubic millimeter of brain tissue was sliced into more than 28,000 layers, each one 400th the width of a human hair. The slices were then imaged and reconstructed into a composite. This painstaking process took 12 days and 12 nights, with the team working around the clock. The resulting map, a single file, 1.6 petabytes in size, captures the essence of a mouse brain section and showcases the beauty and complexity of the brain's architecture. For perspective, this data size is equivalent to 22 years of non-stop high-definition video. And remarkably, the team has already made it available to the public. Subsequently, a team at Princeton University employed machine learning and artificial intelligence tools to trace the contour of every neuron through the slices, coloring the neurons to illuminate them individually in a process known as segmentation. It's important to note that the AI-generated information is still being proofread by the scientists involved in the project. This incredible feat of science has provided a unified view of the mouse-brain connectome, shedding light on how specific parts of the brain are organized and revealing insights into the workings of different cell types. Dr. Forrest Kallman, Associate Director of Data and Technology at the Allen Institute, emphasized the significance of this achievement. One byproduct of this whole project shows us just how incredibly beautiful the brain is, Kallman said in a video shared by the organization. Just looking at these neurons shows you their detail and scale in a way that makes you appreciate the brain with a sense of awe, in the way that when you look up, say at a picture of a galaxy far, far away, he added. This astonishing map, although representing only 1/500th of the full volume of a mouse's brain, contains an incredible amount of information. Within the tiny piece of tissue examined, the researchers identified 84,000 neurons, branched structures that fire off messages down a long arm, called an axon, and then through more than 500 million synapses. Additionally, the map includes 200,000 other brain cells called glia, which provide support and nourishment to neurons. The sheer complexity of the brain is mind-boggling. This tiny piece contained 3.4 miles, 5.4 kilometers, of neuronal wiring, nearly 1.5 times the length of New York City's Central Park. Dr. Nuno Macarico da Costa, an associate investigator at the Allen Institute, highlighted the monumental task of creating the map. It took us about 12 days and 12 nights with the team taking shifts around the clock. It isn't because we were cutting it by hand. It's a machine that is automated, explained Da Costa. We needed to be there to stop at any point in time. If we thought we were going to lose more than a section in a row, if that happened, we would have to start from scratch again, spending another 12 days to arrive at the same point. So it was quite stressful, he said. The creation of this detailed map has provided an invaluable resource for the scientific community, allowing researchers to explore the intricacies of the mouse brain in unprecedented detail. By visualizing and analyzing the connections between neurons, scientists can gain a deeper understanding of how the brain processes information, controls behavior, and develops diseases. Dr. Sebastian Sung, Princeton University S. Evnen Professor in Neuroscience and a Professor of Computer Science, emphasized the significance of this work in transforming brain science. The connectome is the beginning of the digital transformation of brain science, Sung said in a news release. With a few keystrokes, you can search for information and get the results in seconds. Some of that information would have taken a whole PhD's thesis to get before. And that's the power of digital transformation, he said. This achievement marks a significant milestone in the field of neuroscience and has implications for understanding both normal brain function and brain disorders. By unraveling the complexities of the connectome, researchers hope to develop new treatments for neurological and psychiatric conditions that affect millions of people worldwide. The journey to create this map was a long and arduous one, spanning almost a decade and involving the expertise of hundreds of scientists and researchers. The collaborative effort involved a series of experiments and technological advancements that pushed the boundaries of what was thought possible. One of the key challenges was the sheer complexity of the brain itself. The intricate network of neurons and their connections made it difficult to visualize and analyze the data obtained from brain imaging experiments. However, through the use of advanced imaging techniques and the application of machine learning and artificial intelligence, the researchers were able to overcome these challenges and create the first ever three-dimensional map of a mammal's brain. The impact of this discovery is far-reaching and has the potential to revolutionize our understanding of the brain. The map provides a valuable resource for researchers studying brain function and behavior, allowing them to visualize and analyze the connections between neurons in unprecedented detail. This level of detail is crucial for understanding how the brain processes information and controls behavior. Furthermore, the map can be used to study brain disorders and develop new treatments for neurological and psychiatric conditions. By comparing the connectomes of healthy individuals to those with brain disorders, researchers can identify abnormalities in connectivity that may be associated with specific disorders. This information can then be used to develop targeted therapies that address the underlying causes of the disorder rather than just treating the symptoms. One of the most exciting aspects of this discovery is the potential for future applications. The techniques used to create the map can be applied to other parts of the brain, allowing researchers to build a comprehensive picture of the entire brain connectome. Additionally, the map can be used to study changes in brain connectivity over time, such as those that occur during development or in response to injury or disease. Despite the incredible progress that has been made, there is still much work to be done. One of the biggest challenges is the sheer amount of data involved in creating a brain map. The map generated by the research team consists of billions of data points, and analyzing this data requires sophisticated computational tools and algorithms. Another challenge is the complexity of the brain itself, which continues to pose significant hurdles to understanding its function and behavior. However, with continued research and technological advancements, it is clear that we are only just beginning to unlock the secrets of the brain. The 3D map of the mouse brain is a testament to the power of scientific collaboration and the relentless pursuit of knowledge. The researchers involved in this project represent a diverse range of institutions and countries, and their collective efforts have resulted in an achievement that was once thought impossible. As we continue to explore the intricacies of the brain, we can expect to see even more remarkable discoveries that will transform our understanding of ourselves and the world around us. In a world where technological advancements and scientific breakthroughs often seem elusive, the 3D map of the mouse brain serves as a reminder of what can be achieved when brilliant minds come together with a shared passion for discovery. It is a triumph of human curiosity and ingenuity that will undoubtedly pave the way for future innovations in neuroscience and beyond.